Hello, is anyone there?
Yes, we can hear you,
Yes, okay.
Professor.
All right, you can hear me? I didn't
Yes.
see yet. Now I see there's people there. All right. Are there any questions? I saw a lot of emails as I was logging in. I have not looked at my emails yet. So are there any questions before we begin? If not, let me call up our staff. There's the syllabus.
I did have one question, Professor.
Uh-huh.
On worksheet lecture 6, are we just supposed to answer one part of one question as we usually do?
I think so. Let me check on that in just a minute.
All right.
To be certain, but I think so. All right,
Thank
so
you.
there we are. All right, 2-7. So we're supposed to be on chapter 5 and finishing it and starting chapter 6. Let me share my screen. Before I do that, make sure I'm recording. Yep. All right, you should see my screen now. So we should be finishing chapter 5 and starting chapter 6. Realize that we are having a quiz this Wednesday, quiz 3. The quiz is going to cover labs 5 and 6, chapter 4, and the end of chapter 3. It is not covering chapter 5. Any questions about the quiz or what we're going to do? All right, let me go to Canvas. And you said worksheet lecture 6?
Yes, that's right.
Oh, come on. Yeah, only answer 1 or 2 questions from the worksheet.
Okay, thank you.
It says each question has a number, but all questions to each number, except for questions 10, 11, 12, 13, 14. If answering questions 10 through 14, one student answer questions A. And that's probably supposed to be and B. That's got by instead. Let me change that. It looks like a mistake. So A and B. So you'll be answering questions 10A or 10B, and another answer question 10C or 10D. Any question about that?
So to clarify, if it is question 1 and there's question 1, A, B, C, D, we answer A, B, C, and D for 1, correct?
It looks like for question number 1, let me take a look at that I'm asking you to answer question number A, B, 1A, 1B, 1C, and 1D. Well, let me take a look. I'm assuming question number 1 is something that I consider is easier. Let's open that in another tab. Actually, it looks like there's only a question 1A and 1B. I don't see a C, D. 2 has a question 2A, 2B, 2C, and 2D, but I think I'm asking you to answer all of those. Okay, the exception is question 10 through 14. These are more difficult questions. What the heck? That doesn't make sense. Let me take a look at that.
I asked that because I think a majority of us answered just one part of one question each. How do we fix that?
Just go back and if nobody has answered it, just answer it. If not, just grab another question. Okay.
Okay, thank you.
Yeah, this isn't making a whole lot of sense. This. Oh, I see. Why are these two worksheets different? Let me see here. 10 through 11. Well, 10 is correct, but 11 and 12 and 13, 14, I only see one question there. I'm not sure what I'm even doing, but I think we have now about 18 students. So 10, if you're only doing it for 10A and 10B and 10C and 10D, that will be 18, because it doesn't look like there is a question 11. I'm going to guess that I uploaded that and then didn't change it on my webpage, which is really bad. No, that's changed. I'm not sure what I was saying. It looks like only 10 is the one that'll be split. And I shouldn't have used that one because that's older. Yeah, all right. I've got instructions aren't correct. Let me see if I can quickly correct that. I don't want to spend a lot of time on this, but there are the instructions. Here we go. So it looks like only question 10. If that fouls up anybody, just let me know. I don't know where you guys are on questions.
Also, I think what happened is like one person did 1A and then the next person did 1B and then 2A and 2B.
Yeah, try and correct that, because that'll mean not all the questions will be answered. But I
Okay.
understand the problem there, and I won't mark you down if you're doing like 1A and then you do something like 7B, that's fine. The thing is, do try and if you're supposed to answer 2 questions, then do a second question someplace lower down.
Okay, so just pick up where we left off pretty much.
Yeah, just pick up where we left off. All right. Like I said, the instructions weren't correct, and I hopefully that's correct now. Let's go on. Save that. I hope you noticed I did some grading. I'm not going to talk about that. If you have questions, please ask. I don't remember what I graded and what I did not. And I don't want to show you because I'm showing the screen now. So like I said, if you have questions, just ask. I'm going to try and send out your first unknown results tonight. No promises that I'll get it sent out early tonight, but I'll try and do that sometime after the lab, meaning it will definitely be about an hour after the lab. And I will work on that and get that out, but my life has gotten a little complicated lately, so oops, can't close that one. There we go. All right, let's go and see. I've already told you what we're doing, and is chapter 5, and then hopefully we'll start chapter 6. I don't think I've gotten that yet. It looks like we're on slide 45. Yep, there's 2 here. Save that. All right, so we were talking about aerobic respiration, and this slide is showing you a summary of aerobic respiration, starting with glycolysis, shown here, which is followed by the preparatory step in the Krebs cycle, shown here, and NADH and FADH2 from the Krebs cycle, carry the electrons from glucose to the electron transport chain, and this is where the production of ATP happens at a high frequency. And then this slide is also comparing aerobic respiration shown on the left with fermentation shown on the right. Any questions about any of that? I meant to get a chair here so I don't have to kneel. So I'm going to sit here, and if you have trouble, let me know and I'll get closer to the computer. Oh. I'm a little late getting started here. Where's my mouse? Here it is. So the electron transport chain is the third part of aerobic respiration. It's a series of carrier molecules that are in turn reduced and oxidized as electrons from glucose are passed down the chain of the molecules. They are really proteins in a membrane, and the electron is transferred from one protein to another in the membrane. And so they call it like a chain of proteins in the membrane. And that's where the name electron transport chain comes from. The electron is transported through a chain of proteins in the membrane. As the electrons move, they transfer energy from the high energy compounds, starting with glucose, and the energy gets less as the electron moves. And that way the electrons give off their energy in steps, in a stepwise fashion. And that's why when glucose burns in our cells, it doesn't burn at like 1,000 degrees Fahrenheit. It's burning, I don't know what temperature it burns out, but close to 37 degrees Celsius. Energy is released in a process called chemiosmosis to produce ATP. And I've got a little video here, if I can get that to work, showing you an overview of the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Okay, any questions about that? Everyone good? All right. So let's talk about electron transport. Aerobic respiration, the 3rd step is the electron transport. In eukaryotes, this occurs across the inner mitochondrial membrane. So inside the mitochondria in the inner membrane. Among prokaryotes, it occurs along and across the cell membrane. What it is a bunch of proteins in a membrane, and the electrons from glucose, let me blow this up. The electrons from glucose are carried by NADH or FADH2, and the electrons are given here, or the electrons from FADH2 are given here. And the electrons move from one protein to another. And while they're moving, they give some of their proteins, this one, and that one, the energy the protein needs to transfer hydrogen ions from this side of the membrane to that side of the membrane. Any question about any of that? And then where that electron ends up in aerobic respiration, when it leaves the last protein here, the electron actually flows to oxygen. Oxygen and 2 electrons and 2 hydrogen ions get together to form water. Any questions about that? Well, here's a question for you. When you breathe in, what happens to that oxygen? Does anybody know? Anybody want to take a guess?
It's absorbed into the bloodstream.
Yeah, what happens to it though? It doesn't stay as oxygen.
Changes to carbon dioxide.
No, that's a good guess, but no, that's not right. It's actually shown right here. The oxygen you breathe in is converted into water. So all the oxygen you breathe in is converted into water in aerobic respiration. Some of that water will be breathed out. The body actually is pretty good at absorbing that water and then generating water for us, although we're not excellent at it. But desert animals, for example, are really good at converting this water generated in aerobic respiration into water that the animal can use. And I'm talking about something like a desert mouse. Okay. For humans, we breathe out a lot of this water. But that's what happens to the oxygen you breathe in. It's actually converted into water in aerobic respiration. And that happens when the electrons from glucose are carried by NADH to the electron transport chain. And then they move through the chain, and the electron will end up with oxygen. And that is why it's called aerobic respiration, because the final electron acceptor is oxygen. After oxygen, the electrons do not move any further. All right, the other thing happening in this chain is that when the electron is moving through the proteins in the membrane, some of the proteins get enough energy to transport hydrogen ions from this side of the membrane to that side of the membrane. These hydrogen ions are going to want to, by the laws of diffusion, move from this side of the membrane to that side of the membrane. And there's two reasons why they want to do that. First, there's more positive charges on this side of the membrane than on that side of the membrane. So there's an electric attraction for the hydrogen ions to move to this side. The other reason is there's more hydrogen ions on this side than on this side. And then hydrogen will want to move from areas of low concentration to areas of high concentration to areas of low concentration. And so that this hydrogen pumping gradient creates two forces, and then the hydrogen ions want to move this way. There's more positive charges on this side of the membrane. Sorry, my mouse is dying. On this side of the membrane than that side. And then there's just more hydrogen on this side than that side. And so that makes an electromagnetic gradient, and that makes potential energy. When the hydrogen move, they can only move across this membrane in one place. And this is an ATP synthase protein that the hydrogen ions can move down. And when the hydrogen ions move across this protein, it gives the protein the energy it needs to generate ATP from ADP and inorganic phosphate. Any question about any of that? This pumping of hydrogen across the membrane can convert about 40% of the energy in glucose into the energy of ATP. And you might say, let me think about that. Is that right? Make sure I got my numbers right. I think it's 40%. And you might say, well, that doesn't sound terribly efficient, but the best engine that we've ever built, meaning man's ever built, can only convert 20% of the energy in something like gasoline into the energy of motion. And that is the absolute best we can do. So this cell is about twice the best engine. Any question about any of that? All right. Is there anything I need to say about this? I don't think so. So in the electron transport chain, for eukaryotes, we get produced about 32 to 34 ATP molecules per molecule of glucose. Prokaryotes are a little more efficient, and they generate 34 ATP. In eukaryotes, most of our cells can only generate 32 ATP molecules per molecule of glucose. But the heart, the liver, and the kidneys are more efficient, and they can generate 34 molecules of ATP. Why might the heart, the liver, and the kidney be more efficient than, say, something like your nose cell or your skin cell? Or in this case, your brain cells. Why do you think the heart in particular is more efficient than your skin cells?
Is it because they pump blood in and out?
Yeah, the heart, the liver, and the kidney are always working. And so they're more efficient than other cells in your body. Like your muscle cells, they only work hard when you're, exercising or running or something like that. Most of the time, they're not working hard. And so the heart, the liver, and the kidney, they're always working. The heart, as you know, is always pumping blood. The liver is always purifying the blood. I'm not going to talk more about that. And then it's when you're eating a meal, it'll take the glucose out of the blood and store it in the liver. And then when you're not eating and the blood needs more glucose, it'll take the glucose from the liver and pump it into the blood. And then the kidney is always purifying the blood and making urea to purify the blood. But anyways, these three organs are always working, so they're more efficient than all the other cells in our body. And they can generate 34 ATP per molecule of glucose, but all our other cells only generate 32. And I've already mentioned the final electron acceptor in aerobic respiration is oxygen. It combines with hydrogen and the electrons to form water. This is showing you an overview of the aerobic respiration. And this is showing you what happens with NADH. Let me blow that up. Actually, I think I've got it better in this slide here. NADH bringing in the electrons, and then the electron transfers to this protein, to that protein, to that protein, to that protein. Oops, sorry. To this protein, to that protein, to that protein, to that protein. This is the last protein. The electron then flows out of the electron transport chain, and it flows to oxygen, which is converted into water. And the point is, the electrons give up their energy in steps, and the release of energy means the cell can stay at about 37 degrees instead of 1,000 degrees Fahrenheit, which would kill the cell, by the way. And so the energy in the electrons is reduced slowly in steps. And this is a picture of the inner mitochondrial membrane where ATP production occurs in the electron transport chain, the mitochondrial outer membrane, and then the inner membrane. And the electrons are brought by NADH on this side of the membrane, so that's the outside of the inner membrane. And then the electrons flow through the proteins in the chain. The hydrogen ions are pumped to this side of the membrane, and how they are protected from, meaning they don't damage the cell, is that this is the space between the inner membrane and the outer membrane. And so that's where the hydrogen ions are. And then they flow through the ATP synthase here to make ATP. So in eukaryotes, that's happening in and across the inner membrane, and then the hydrogen ions are pumped between the inner membrane and the outer membrane. For prokaryotes, this happens in the cell membrane, and it's the same proteins in a membrane. It's just the cell membrane instead of the inner membrane. And then the hydrogen ions are pumped from this side of the membrane to this side of the membrane. Where they are here is in the cell wall. Okay. Any question about any of that? All right. The hydrogen ions, of course, can move freely in the cell wall, and they will generally just stay in the cell wall until they move down the ATP synthase and then make ATP and water. Any questions about any of that? The redox reactions happen. Let me see if I can show you that. And that's this step here. And I didn't mention it, but there you can see when the electrons move to this molecule, it will be reduced, gain of electrons, GER, reduction. And then the molecule will give it to Q, the next molecule down. And so this molecule will become oxidized and Q will become reduced. And that's the redox reactions as the electron moves from one molecule to the other. I already mentioned that the electrons create a greater hydrogen ion gradient out here. This is called the proton motive force. It's really potential energy. And the hydrogen ions are here. They want to come over here. They can only move through this channel protein, which makes ATP. And the cell uses this energy difference to make ATP. This is called chemiosmosis. I don't usually use that term, but the flowing of hydrogen ions makes ATP by chemiosmosis. Another name for the electron transport chain is oxidative phosphorylation. I don't tend to use that word. It's called oxidative phosphorylation because the proteins in the membrane will become oxidized, and then ATP gets made, meaning ADP becomes phosphorylated. Phosphorylation is just the adding a phosphate to a molecule, and phosphate is added to ADP, and that's where this term comes from. You don't really need to know any of that, and I don't use this term. I always use the term electron transport chain. There's actually 4 names for this, and I always use electron transport chain. But some people use electron transfer chain, and that's because the electron transfers from one protein in the chain of proteins in the membrane to the other. Students don't generally get confused between electron transfer chain and electron transport chain, but I'll use the term electron transport chain. Any question about any of that? I think your book uses the same terms I use, by the way. All right, so in aerobic respiration, when we add in the ATP made in glycolysis, the preparatory step and the Krebs cycle, and then the electron transport chain, for prokaryotes, we make 36 to 38 ATP molecules. The 38 ATP being made in the heart, the liver, and the kidney cells. All the other cells make only 36. And then prokaryotes are a little more efficient. They make 38 ATP per molecule of glucose. Any question about any of that? And this is the summary equation to explain aerobic respiration. You get 1 glucose, C6H12O6. You add on 6 oxygen molecules and 38 ADP molecules and 38 phosphate. The I just stands for inorganic phosphate. And that generates 6 CO2, 6 water molecules, and 38 ATP molecules. The water, all of this comes from all of the water comes from oxygen. So 6 times 2 is 12, and then 6 times, oh, this isn't a balanced equation. This should be 12 oxygen, and we add on 6 water here. You don't need to know the balanced equation, but all of the water comes from oxygen. All of the CO2 comes from the burning of glucose. And the burning of glucose happens in the preparatory step and the Krebs cycle. So all of the carbon in glucose is burnt into CO2, and none of the CO2 comes from the oxygen we breathe in. All of it comes from glucose. Any question about any of that? And remember, in aerobic respiration, the electrons from glucose flow to oxygen, which will be converted into water. So this is showing you aerobic respiration. In summary, all the different steps. We generate 2 ATP in glycolysis, 2 in the Krebs cycle in the preparatory step, and the 2 are made in the Krebs cycle. And then 34 to 36 in the electron transport chain. Did I get that right? No, 32 to 34 in the electron transport chain. Any question about any of that? Oh, there it is there. 2 from glycolysis, 2 from the Krebs cycle, and then 32 to 34 from the electron transport chain. It's showing you for prokaryotes why it's only got 34. All right. So that's aerobic respiration. Let's compare it to anaerobic respiration. Anaerobic respiration is similar to aerobic respiration. One major difference is anaerobic respiration does not have the electrons flow to oxygen. The electrons flow to another molecule other than oxygen. So we say the final electron acceptor is another molecule other than oxygen. And it depends on what form of anaerobic respiration it is. It could be nitrate, shown here. It could be nitrite, that's NO2 with a negative charge. It could be sulfate. It could even be iron, and we talked about iron before. It just depends on what chemical reaction the microorganism does. Generally, anaerobic respiration includes glycolysis and the Krebs cycle. The only difference is that the electron transport chain is present, but it is shorter than the electron transport chain in aerobic respiration. So let me show you the chain here. There it is there. It's generally shorter. And because it's shorter, we generate less ATP for anaerobic respiration than for aerobic respiration. It depends on which anaerobic respiration we're talking about, how much shorter it is. me go back to where I was. There we go. So generally, our anaerobic respiration includes both glycolysis, the preparatory step in the Krebs cycle, and then the electron transport chain. But the electron transport chain is a little different. For one thing, it's shorter than an aerobic respiration. And then the electrons do not flow to oxygen. They go to some other molecule. The ATP yield varies depending on which form of anaerobic respiration it is, but the total yield of ATP for anaerobic respiration is between 2 ATP per molecule of glucose and between 36 molecules of ATP per molecule of glucose. So the very best, the 36, are very close to the prokaryote, which can generate 38. And then the worst are very close to the ATP production in fermentation, meaning the very worst is close to 2. Any question about that? All right, because anaerobes are not as good at generating ATP from a molecule of glucose compared to aerobes, the anaerobes tend to grow slower than the aerobes. So aerobes grow the quickest, and anaerobes tend to grow slower than an aerobe. Any question about any of that? All right, if not, that's it for anaerobic respiration. Let's talk a little bit about fermentation. We've shown you this slide showing you, actually in this slide, it's showing you just respiration on the left. We used it to talk about aerobic respiration, but generally all forms of respiration are similar, and that is they have glycolysis, the preparatory step, and the Krebs cycle, as well as an electron transport chain. Fermentation is shown on the right, in which case glucose will change to pyruvic acid in the normal way in glycolysis. And then the pyruvic acid will change to the fermentation end product. And there's more than one fermentation end product because there's more than one fermentation reaction. But the two that we'll talk about are where ethanol, ethyl alcohol, is the fermentation end product. And that's the one we use to make beer and wine. I think that's the one that's used also, at least in part, to make fermented breads, like sourdough bread, something like that. And then the glycolysis is followed by 1 or 2 fermentation steps. Let me blow this up. So it depends on the fermentation step we're looking at. It looks like this is the normal fermentation. What the heck is that called? Sorry, I'm drawing a blank here. It's not alcoholic, lactic acid fermentation. This is what it looks like we're looking at here. And I'll talk about that in a minute. A fermentation releases energy from the oxidation of an organic molecule. Usually, this happens with glucose. And glucose will usually be made by glycolysis and then released into the blood. But glycolysis proceeds as discussed, and it'll generate 2 pyruvic acid molecules, a net gain of 2 ATP molecules, 1 NADH, and then 1 H plus from the oxidation or fermentation of glucose. Glucose is the preferred fermentation product, but like aerobic respiration, other food molecules can be used. Certainly any carbohydrate can be, and usually the other carbohydrates will be converted into glucose and then sent down the aerobic respiration chain or process in the normal way, the glucose way, which we discussed. In glycolysis, we generate NADH also. What happens to the NADH happens in the next step. Following glycolysis, we have the fermentation steps, the directly fermentation steps. And this is only looking at, well, this isn't looking at anything. So it's usually 1 or 2 chemical reactions after glycolysis to finalize fermentation. Fermentation does not require the presence of oxygen, so it can happen when oxygen is around, or it can happen when oxygen is absent. Fermentation does not use the Krebs cycle, at least in the changing or destruction of the molecule to generate glucose or glucose 6-phosphate. Fermentation does not use the Krebs cycle, does not use the preparatory step. And then where the electron flowing from glucose ends up is the organic molecule, which in this case is pyruvic acid. The electrons flow to pyruvic acid. And so that is the final electron acceptor in fermentation. Electrons flow from glucose to pyruvic acid, and the pyruvic acid then uses that electron for its purposes. Largely, it makes the molecule charged. NADH that was generated in glycolysis, in the fermentation step, the NADH is converted into NAD+, which will recycle the NAD+. Why do you think your muscle cells will undergo fermentation when you're not getting enough oxygen in your blood from your breathing? So that's why when we're jogging or running, we will start breathing heavily. It's because of the carbon dioxide building in your system. What's the point I'm trying to say here?
Isn't that something that your body needs to get rid of, the lactic acid?
Yeah, the lactic acid is. I'm not talking about that. I'm talking about the NADH. Why does our muscle cells use up the NADH in the fermentation step to generate NAD+? Why does the muscle cell use this up in fermentation instead of just keeping it around in the muscle, the NADH? Because if the cell were to keep NADH in the muscle, eventually the heart and the lungs will deliver blood to the muscle cell, which will deliver oxygen, and then the cell will stop using fermentation. I'm talking about our muscle cells, and then it'll switch over to aerobic respiration. Okay, does anyone see why we use up NADH in fermentation? It's actually shown here. All right, the reason why the muscle cell wants to use up the NADH, because when it does, it'll regenerate NAD+, and as long as the muscle cell has NAD+, the NAD+ can allow glycolysis to happen. If there is no NAD+, glycolysis will stop happening, And if glycolysis ever stops happening, there will be no production of FTP by glycolysis. Any question about any of that? All right. There's 2 fermentation reactions you need to know about. There's something like 5 in totality, but glycolysis happens in both of the fermentation processes. And then after glycolysis, we have a fermentation step or steps. In lactic acid fermentation, we run glycolysis in the normal way. And then pyruvic acid is converted into lactic acid. And in the process, NADH is used up and converted into NAD+. And if you notice, we need to have NAD+, because if there's none of it in the cell or the mitochondria will stop making ATP. And why the cell does this is that if there is no NAD+, then we will stop glycolysis. And if there is NAD+, we can continue using glycolysis for as long as we have glucose or NAD+. Any question about any of that? Anyway, that's lactic acid fermentation shown on the left. We use lactic acid fermentation to make most of our cheeses, like cheddar cheese, but not all cheeses. We use the lactic acid fermentation to make a lot of our fermented food, like sauerkraut. I'm drawing a blank on some other fermented foods. Chocolate uses lactic acid fermentation to ferment the chocolate. And I think black tea is also fermented, and I think it's lactic acid fermentation. So those are some of the fermentation reactions that occur with the cells. Lactic acid fermentation is the second reaction that humans have to generate ATP, meaning all of the decarboxylation reactions, other than aerobic respiration and lactic acid fermentation, do not generate ATP. This is showing you on the right another fermentation reaction, and it's a 2-step reaction where pyruvate is converted into 2-acetylaldehyde, and this is a 2-carbon molecule. So one of the carbons has to burn off as CO2 before this reaction can proceed. And then 2-acetylaldehyde will be converted into ethanol by NADH. Any question about any of that? All right. There are different fermentation end products depending on what fermentation reaction we're talking about. And this is showing you the different fermentation reactions. There's something like six of them. Our cells can only engage in one fermentation reaction, and that is our muscle cells. They can engage in fermentation. Otherwise, all of our cells have to have iron or cast iron or stainless steel, something like that, for the reaction to proceed. So lactic acid fermentation happens by a number of organisms. We tend to use most of these organisms for making yogurt. Ethanol or ethyl alcohol fermentation was given to my friend who teach biology mostly, and yeast engage in ethanol fermentation, and they make ATP from the pyruvic acid, which will be, the pyruvic acid will be converted into ethanol. Any question about any of that? There's Propion bacterium, which makes propionic acid. This is the fermentation reaction that is used to make Swiss cheese. If you don't know, Swiss cheese is different from most other cheeses because most other cheeses use lactic acid fermentation of the milk, shown here. And alcoholic fermentation happens mainly by yeast, which will ferment the sugar and convert it into alcohol. There are different fermentation reactions and they make different products. Have I talked about this one? I'm trying to remember. Yeah, I talked about this one. Propionic acid is a 2-carbon molecule, and pyruvate is a 3-carbon molecule. So one of these carbons has to be burnt into CO2 right away, or the fermentation reaction cannot proceed. My point of the CO2, when that CO2 is given off, that creates the holes in Swiss cheese. Swiss cheese is not made by lactic acid fermentation. It's made by propionic acid fermentation. Any question about any of that? All right, there are other fermentation reactions that can happen. We're not going to talk about them, but there are different organisms like Clostridium, E. coli, Enterobacter. They can engage in different fermentation reactions. Like for Enterobacter, it can engage in ethanolic fermentation, but it can also engage in lactic acid fermentation. And what you do with lactic acid fermentation is you get out of the turn lane and get on the main highway. And then when it's time to stop, you can ask for whatever your car uses to ferment the car or ferment the fuel in the car to get the car to run. Any questions about any of that? All right, so this is showing you in a nutshell the different fermentation end products. It's industrial or chemical use, which I won't ask you about that. And then the sorting. So ethanol made by a beer or spirit company, meaning wine, can be converted into malt extract. That's what it starts with. And then that will be converted into ethanol by fermentation. And really the only organism that does this is Saccharomyces viridans, I don't remember a fungus. The yeast does this one, and maybe a fungus. Wine doesn't use lactic acid fermentation, which is above. Excuse me, that's alcoholic fermentation. Wine does use this fermentation. You start usually with grape or some other fruit juice, and then the yeast engage in alcoholic fermentation, making ethanol. Lactic acid fermentation is the one we talked about, and it makes vinegar. Excuse me, that's not lactic acid fermentation. It makes yogurt and cheese, rye bread, sauerkraut, summer sausage, and most of our fermented foods happen by lactic acid fermentation. The starting substance can vary for that one. If you're going to make yogurt, you start with milk. For rye bread, you start with the grain of rye or with sugar, or usually both. Sauerkraut, you start with cabbage. And then you use an organism to ferment it. I'm not going to quiz you on that. All right, any questions about any of this slide? The important part is there's different fermentation reactions, and in industry, we tend to use them to make specific substances for ourselves. Like if your industry needs acetone or butanol, you can actually make it from the fermentation of the sugar maltose. All right, if there's no questions about this, I'm going to move on. This slide is showing you the end product, meaning the last position that the electrons flowing from glucose flow to. And the point is, in aerobic respiration, I can't read that now, which is here. The final electron acceptor is oxygen. We generate 36 to 38 ATP, with prokaryotes being 38 molecules of ATP per molecule of glucose. Anaerobic respiration differs. It does have the preparatory step, the Krebs cycle, and glycolysis, as well as electron transport chain. But this electron transport chain is different. For one thing, it's shorter, has fewer proteins in it. And then the production of ATP can vary. It can be anywhere between 2 and 38 molecules of ATP. Fermentation is a little different. Fermentation can be either anaerobically or aerobically, but in both environments, fermentation can happen. And the reason for that is that the bacteria, or whatever the yeast that's doing the fermentation, can ferment the ingredient we're adding in. No oxygen is needed, so no oxygen is made. And then the amount of ATP that's made by fermentation is 2 ATP molecules per molecule of glucose. Any question about any of that? All right. Now I've talked about glucose being used in aerobic respiration, and most of the time that is the molecule that we use when we want our cells to make ATP from aerobic respiration. However, all of the food molecules that we can eat can be used to generate ATP. Carbohydrates usually are just converted, meaning you remove the last sugar, and then you add water to it, and that comes into play by the what is that called? The not dehydrogenation, the hydrogenation of water. So not only glucose is used, not only carbohydrates, and carbohydrates would be broken down into glucose, but all food sources may be used. There is a problem for the large molecules, like a large molecule of fat, or a large protein, or a large carbohydrate. And the problem is the cell that the peanut butter would like to move through will not allow the peanut butter to move through. And that's the case with peanut butter, which is a solid. It's not the case for peanut butter that's not a solid. Anyways, the problem with this is that the large molecules, like the large protein molecule in peanut butter, cannot get through the cell membrane. The cell solves this by making a transport protein that can then move the large molecules outside the cell into the cell, or the cell will dump enzymes, digestive enzymes outside the cell membrane, and then those enzymes would start dissolving the protein, the enzyme. Any questions about any of that? All right. Another way that the cell can utilize these large molecules outside the cell is to secrete lipases and proteases extracellularly, meaning you're secreting them outside the cell, and that will break down lipids and proteins outside of the cell, making them into smaller components. With the the protein, the extracellular proteases will digest the protein into amino acid. Amino acids are now small enough to enter the cell, although they do that in a transport protein if they're a eukaryote. And this is the way that humans actually digest their large lipases and proteases. We digest the enzymes into our gut and then allow the enzymes to digest the protein in the ham, for example, or any other protein. I'm not sure I want to talk about this. We'll mention briefly that amino acids, when they're digested, meaning the protein will be digested into amino acids, the amino acids can be utilized and brought into the cell. The trouble with the amino acids is they have an amino group, that's NH2 if you remember, and that has to be removed before we can take this protein and then digest it by the aerobic respiration pathway. So the protein can be digested into amino acids outside the cell. The protein can be digested outside the cell, and then we continue with what we happen to do with like lactic acid fermentation. You convert that protein into lactic acid. I sort of diagrammed it, and I'm not talking about it here, but let's go back. There we go. The electrons flow from glucose to pyruvate, which will then be converted in lactic acid to lactic acid. And then in the making of vinegar, the electrons will flow to the paper towel underneath wherever it's being held or what. All right, let's return to where we were. The point is that these large molecules can be digested and used in aerobic respiration or another form of respiration. They can also be used in fermentation. If they're used in fermentation, then we will only get 2 ATP molecules, whereas aerobic respiration, we get many times more. All right, any question about any of that? All right, I've talked about in this lesson treating carbohydrates, lipids, and protein as separate and distinct molecules. They are not totally separate, meaning there are crossovers, and animal cells are very bad at crossing over and making biosynthesis, making a larger molecule out of smaller molecules. But I already mentioned the carbohydrates can be utilized by aerobic respiration by converting the carbohydrates to a sugar and then converting the sugar into glucose and then sending glucose down the aerobic respiration pathway in the normal way. Any questions about any of that? With lipids, they can be digested and burned in aerobic respiration. The lipids will be disassociated into fatty acid portion and then the glycerol. Remember, the glycerol is holding the fatty acids together in the molecule of lipid. Proteins too can be digested, but let me talk about lipids first because they're a little easier. Lipids will be changed into glycerol, and then glycerol will enter the Krebs cycle about midway down. And then the carbon in the glycerol will be burnt into CO2. And ATP will be generated out of that. Fatty acids will break off the lipid, and then the cell will break these fatty acid into 2 carbon links. And then these 2 fatty, what used to be a fatty acid, the 2 carbon fatty acid will come into the Krebs cycle at the start. And then each of those fatty acid, the carbons in the fatty acid, will be burnt into glucose. Any question about any of that? Generally, all the carbohydrates, the lipids, and the proteins will be metabolized with glycolysis, the preparatory step in the Krebs cycle, and then the electron transport chain. But occasionally, it will happen very, very late in the chain. I used to know this when we digest proteins and engage in aerobic respiration, some of the amino acids enter the Krebs cycle very late in the cycle. Some of them enter early, and that is as they will be converted into pyruvic acid, which is the last member of the it's not catalase, the Krebs cycle process of aerobic respiration. Any question about any of that? All right, and then the electrons will flow in both cases, or all cases, to the Krebs cycle, and then will flow to the electron transport chain. And those electrons will be used to pump hydrogen ions across the membrane. But hold on to more or less the hydrogen ions. Otherwise, we would always be losing hydrogen. All right, any questions about this? When we're talking about the different methods to generate ATP, aerobic respiration is the most efficient. In eukaryotes, aerobic respiration happens with 32 to 34 ATP per molecule of glucose. Other metabolic pathways for generating ATP are not as efficient, and so they will generate less ATP. Any questions about any of that? All right. I've talked about each of these molecules that I've discussed today as being independent and not really related, meaning if one molecule up on the chain gets blocked for whatever reason, the molecules lower in the chain can proceed as long as there's molecules lower in the chain. Eventually, we'll run out of molecules, and then they will be blocked wherever molecules are blocked. I'm not sure, probably in the liver. But there are common intermediates between both anabolic and catabolic pathways, and then there are common intermediates between catabolic pathways in humans. we call the metabolic pathways that can function both ways, meaning normally it will function this way in aerobic respiration, but when another reaction is occurring or something funny is happening with aerobic respiration, and it's not glucose we're sending in like a lipase, and it results in changes, and then the cell will need to get ATP back, and so it'll go this way. So the generating of ATP goes this way, and we'll utilize, like you got a cake, we'll utilize it and go this way. And then if you ferment your beer or something like that, the reaction will go that way. Any questions about any of that? The point being, these pathways, we call them amphibolic because the metabolic pathways can function going both ways. Wherever we see a double line, the reaction can go both ways. And that's why, although this isn't shown up here, this should be a double way. I think this is an abbreviation for another slide. And this is another one. When we make lipids ourselves, we'll combine fatty acids with glycerol, and then the glycerol and the fatty acids will form the lipid. Here's a question for you. Suppose we eat a high-fat diet. Will that fat be digested, or will some of that fat be converted into fat in our belly? Anybody? Nobody's going to
Depends on
say?
what you eat.
Go ahead.
Depends on how much you eat of it.
Yeah, if you're eating too much of a rich, fatty, a fat diet, you're eating too much and you're not eating much carbohydrates or proteins, what would happen to those lipids? Would you store them down as body fat? Nobody's going to take a guess? You would. Obviously, if you eat too much of anything, you'll start storing it as body fat. But if you eat too much lipids, most of us couch potatoes, the lipid will be broken down. And that's not shown here, but the lipid being broken down into glycerol and fatty acids, that can go both ways. The glycerol will then be converted into a the glycerol will be utilized by the glycolysis and will enter right here. Sorry, my mouse is dead. Come on, mouse. Oh, there we go. And then the fatty acids will be broken down into 2 carbon chunks and will enter the Krebs cycle here. However, if we eat a high-fat diet and we don't need ATP, your body will go this way, reverse. The 2 carbon units of the fatty acid will be combined with the glycerol, and then this molecule goes backwards. Like I said, this is double link right there. And I got to get that out of there so I can see this. And we will go backwards, and we could make glucose. We could also go backwards and then generate glycerol and fatty acids and then remake the lipid. Okay, and that's what a lot of us do. Us couch potatoes, when we eat a high-fat diet, if we're not burning off those energy, we will generate lipids and then store it down as body fat. If you did need a protein, some of this lipid could be converted into proteins, and there would be making that amino acid here. This would be making this amino acid here. That would be making that amino acid here. And that would be that making that amino acid here. And the coenzyme A's could easily make the amino acids. They could also go this way to make the amino acids. And then the glycerol will come this way to make the amino acids. Most of our couch potatoes would do that, digest the lipid, and then use it to make a fat, and then we'll store that down on our belly as a lipid. Any question about any of that? Okay. And if we didn't need glucose, some of those lipids in the high lipid diet could be converted into glucose. And then if you needed a protein, we could convert some of the fatty acids into amino acids and then build up the protein from the amino acids. Humans are not very good at going both way in their reactions, but with these reactions, they can go both ways. And green plants are much better at going both ways. So they're more amphibolic than like humans, for example. Any question about any of that? If not, let me end that and talk briefly. We're after our time. So I'm going to stop here, and I'll see you at 6:30 for the lab. Oops, there.